Hello, everyone, and welcome to today's live broadcast, Importance of Sample Preparation and Choosing the Right Gel for SDS-PAGE. It is presented by Alok Tomar, PhD, Product Manager for Protein and Cell Analysis at Thermo Fisher Scientific. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about our sponsor, visit www.thermofisher.com. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Tomar. I will now turn the presentation over to him.
Hello, everyone, and welcome to this gel webinar where we will review sample prep tips and go over key differences in precast gel chemistries that can affect migration, straightness, sharpness, and overall performance of your protein gel. These are the ingredients that are required to achieve an optimal gel electrophoresis. Well-prepared samples, high-quality protein gels, choosing the right chemistry for your target application and proteins, using the recommended buffers for that particular gel chemistry, using the correct gel running apparatus, and following the recommended gel running protocol, which includes information about current or voltage and running time. The first part of this talk is about sample preparation or sample prep, in short, for gel electrophoresis. Poor sample prep has known to produce results that are difficult to interpret. The sample prep methods vary for different sample types, and sample prep methods can introduce incompatible substance for gel electrophoresis. This is the list of most common offenders when it comes to sample prep. Excess amount of salt, detergent, and lysis buffer will have a noticeable effect on gel electrophoresis. DNA or nucleic acid contamination, the type of sample loaded and amount of protein loaded, the use of excess or incorrect reducing agent, loading different amount of samples in adjacent wells, having empty wells adjacent to sample wells, and overheating of samples can all have adverse effect on gel electrophoresis. Okay, so let's start with what happens when you use excess salt. On the right, the gel image shows the effect of increasing sodium chloride or NaCl salt concentration on electrophoresis. Now, lane 1 is the unstained ladder, and the lanes 3, 5, 7, and 9 are 10 microgram of E. coli lysate with increasing amount of sodium chloride salt concentrations. The 500 millimolar and 1 molar NaCl concentrations, they result in uneven sample lanes and lane widening, which is particularly noticeable towards the bottom of the gel. The high salt concentrations causes increased conductivity during electrophoresis that affects protein migration and can result in gel artifacts in adjacent lanes containing samples with normal salt concentration to the extent that the data from such lanes might be unusable. If your sample has high salt concentration, either because of the lysis buffer composition or some other reason, you might have to perform dialysis to lower the salt buffer concentration, or you can precipitate your samples and resuspend them in lower salt buffer prior to electrophoresis. Make sure that the salt concentration does not exceed 50 to 100 millimolar final concentration. Protein precipitation by ammonium sulfate is a commonly used technique. Similar to the gel on previous slide, using 10 microgram E. coli sample with increasing concentration of ammonium sulfate concentrations in lane 3, 5, 7, and 9 resulted in viscous samples that formed streaks at sample lane edges. So you can see that the bands are not even, like in lane number 3. Specifically in lane number 9, you can see these prominent streaks towards the edges of the lane, and that is being caused by higher viscous sample because of this higher salt concentration. There is also lane widening. This is also particularly more visible towards the bottom of the gel. The possible causes and solution are similar to what we described in the previous slide. For Western blotting applications, it is common to use a lysis buffer with 1% SDS concentration. This can be sometimes higher depending on the sample type. In a diluted sample, when higher volume is loaded per well, the total SDS concentration can significantly increase. The total SDS concentration is 2 and 4% respectively in lanes 3 and 5 in this gel that is on your right-hand side. Increasing SDS concentration results in lane widening and uneven sample lanes. As you can see, lane number 5 is much more wider compared to lane number 3, and there is twice as much SDS concentration in lane 5. So these are the artifacts caused by having increased amount of SDS in your samples. Similarly, Triton X100 is also a widely used non-ionic detergent for lysing cells to extract proteins. RIPA buffer, which is a commonly used lysis buffer, can have 1 to 2% Triton X100 concentration in it. The gel that you're seeing on your right-hand side, lanes 7, 8, and 9, they have increasing amount of Triton X100 concentration. And as you can see, with increasing concentration of Triton X100, the lanes are getting more wide. They are losing some resolution towards the bottom of the gel, and they are uneven. The cause of such effect on gels is because detergent from mixed micelles with the anionic detergent, which is SDS, which is present in the gel and which migrates down into the gel, this non-ionic detergent can interfere with the SDS protein binding equilibrium. Most of the non-ionic detergents like Triton X100 are worst when it comes to interfering with SDS-PAGE. So a rule of thumb for SDS-PAGE applications is to keep the ratio of SDS to non-ionic detergent at 10 to 1 or greater to minimize this artifact of having to disrupt the equilibrium of SDS and protein binding. The solution here would be obviously to remove the excess detergent, and for this, detergent removal columns are available, or you can use SDS-PAGE sample prep kits that could help remove this excess detergent from your samples. Now, RIPA, or radio immunoprecipitation assay buffer, is a common lysis buffer. The recipe for RIPA buffer used in this particular experiment is provided in the right bottom corner. The RIPA buffer has salts, detergents, reducing agents, which can all affect quality of gel run if the final concentration of gel increases for any reason. As demonstrated on this gel on the right, that increasing final RIPA buffer concentration up to 4x causes lanes to flare out at the bottom, resulting in wider lanes and streaking. This is caused by increasing the levels of salts, detergents, and reducing agents in the lanes, which are all known to affect quality of the gel run. The solution is to concentrate the samples in RIPA lysis buffer before loading them onto the gel to avoid having to load higher levels of this buffer per well. Now, DNA contamination can have a significant impact on SDS-PAGE. The gel demonstrates that protein samples contaminated with 1, 3, or 5.6 mg per ml in lanes 5, 7, and 9 respectively causes significant protein clumping, which results in narrowing of the lanes and visible protein streaks during electrophoresis. DNA contamination causes the samples to become very viscous that can result in protein clumping and streaking during electrophoresis. Sonication and other methods that help shear genomic DNA can help reduce viscosity and can minimize the impact of DNA contamination on gel runs. Now, loading wells with 20, 30, or 40 microgram of total protein concentration resulted in bands losing resolution, with streaking observed in most of the lanes. At 10 microgram of total protein concentration per well, the E. coli lysates in lane 2 or rat liver lysate in lane 7, they produce sharp, well-defined bands with good resolution. Increasing rat liver lysate loads in lanes 8, 9, and 10 has much more prominent effect on band resolution and streaking compared to E. coli lysates. This demonstrates that maximum amount of protein loaded per lane will depend on your sample type. However, as a general rule, it is recommended to not go over 0.5 microgram per band, or about 10 to 15 microgram of cell lysate per lane. So if you're using a purified protein, for example, you should not be loading more than 0.5 microgram per well, because all of that is going to be concentrated in one band, which is going to be a very prominent band. But on a cell lysate, because you can have basically hundreds of bands, again, every band should not be more than 0.5 microgram, which is usually the case. So the total protein lysate, when you are using a wholesale lysate, whether it be cell or tissue, the limit would be about 10 to 15 microgram per well. So reducing agent can be part of lysis buffer, sample buffer, and gel running buffer to keep proteins under reduced state during electrophoresis. As demonstrated in the gel on the right, increasing the concentration of DTT to 50 millimolar in lane 5, 500 millimolar in lane 7, and 1 molar in lane 9 results in narrowing of the lane and creates a shadow at lane edges. It is recommended that the final concentration of reducing agent should be less than 50 millimolar for DTT and TCEP, and should be less than 2.5% total for beta-mercaptoethanol. Now, different reducing agents can have different impact on protein integrity combined with the heating temperature and time. The image on the left is the new page Bis-Tris gel from Invitrogen with various sample prep conditions. The lane 2 has recommended sample prep condition for new page, which includes mixing the sample with new page sample buffer and addition of 1x new page reducing agent, which is essentially DTT, and boiling the sample at 70 degrees Celsius, not 100, but 70 degrees Celsius for 10 minutes. This provides optimal protein denaturation and reduction for Bis-Tris gels. Under this optimal sample prep condition, as demonstrated in lane 2, there is very minimal sample degradation and protein integrity is maintained. However, use of XTRA, which is the Bio-Rad criterion XT reducing agent, instead of DTT in lane 3 and boiling the samples for 95 degrees Celsius for 5 minutes, as recommended by Bio-Rad sample prep protocol, results in significant protein degradation. The use of Tris-Glycine sample buffer with 50 millimolar DTT and boiling the sample for either 95 degrees Celsius for 5 minutes or 85 degrees Celsius for 2 minutes reduces sample degradation significantly, as shown in lane 5 and 6 respectively. The image on the right shows that the effect of sample prep on protein integrity when using a Tris-Glycine gel. Compared to Bis-Tris gel, the Tris-Glycine gel shows more degradation, but the choice of sample buffer, reducing agent, boiling temperature, and boiling time has a big impact on protein integrity and degradation levels. TCEP, or Tris-2-carboxyethyl phosphine, is the reducing agent present in Bio-Rad criterion XT reducing agent. This slide shows different prep methods using TCEP and its impact on sample integrity. Lane 2 has the recommended protocol for new page Bis-Tris gel with new page sample buffer and 1x TTT, followed by boiling the sample at 70 C for 10 minutes. Lane 7, 8, and 9 demonstrate that heating the samples in presence of Bio-Rad XT reducing agent, which contains TCEP, results in significant protein degradation. This slide demonstrates that reducing agent TDP, or tri-N-butyl phosphine, at different concentration is equally effective as DTT in preserving sample integrity. However, again, Bio-Rad XT reducing agent in lane 9 results in significant sample degradation. So choosing the right gel and the right protocol for reducing your samples and denaturing your samples is going to play a vital role in how much you are able to preserve your precious samples and avoid sample degradation. This is a summary slide for all of the sample prep data that we saw today. You will have access to this webinar, and this slide will help to provide an overview of issues associated with sample prep and their potential solutions. This is protein gel electrophoresis 101, and now that lists the frequently used types of protein electrophoresis. As you know, PAGE stands for polyacrylamide gel electrophoresis. In a native PAGE application, ionic detergents like SDS are not added to any of the buffers, resulting in the charged proteins being separated according to their native physical properties in an electric field. SDS-PAGE, or sodium dodecyl sulfate PAGE, SDS is an ionic detergent, and when added to gel electrophoresis buffers, it denatures proteins and binds to these denatured proteins, providing a uniform negative charge to all protein, which facilitates separation according to size only. As shown in the schematic to the right, the SDS detergent in presence of reducing agent can separate the two subunits of a protein, which are held together by disulfide bonds. Reducing agent like DTT or beta-mercaptoethanol can reduce disulfide bridges to achieve better denaturation. The negatively charged SDS molecule then binds uniformly to entire protein, which results in separation according to the size of the protein. The smaller subunit A migrates faster than the biggest subunit B, thereby creating a size-based separation on the slab of polyacrylamide gel. Okay, so in the next few slides, we will review the different gel chemistries that are available in precast gels and their specific advantages and disadvantages. The two most widely used gel chemistries are Tris-Glycine, which uses traditional lamelli buffer, and Bis-Tris chemistry. The other two specialized gel chemistries are Tris-Acetate gels for separation of large molecular weight proteins up to 500 kilodalton, and Tricine, that is specialized for separation of peptides and small molecular weight proteins as small as 2.5 kilodalton. Let's take a quick look at the buffer system of gel electrophoresis. The continuous buffer system utilizes the same buffer in the gel and the gel running buffer. The discontinuous system, which is utilized by most commercially available gels, has a different gel buffer and the gel running buffer. It has at least 2 gel layers of different pore sizes and has different buffer composition for the stacking gel, which condenses the protein sample into a nice tight band during electrophoresis, and the separating gel, which is responsible for creating the size-based separation of protein samples. This slide provides a quick summary of the discontinuous buffer systems utilized by the 3 most popular gel chemistries. The Tris-Glycine system includes Tris and chloride ions in the gel buffer, which has a pH of 8.7. The running buffer ions are Tris, glycine, and ionic detergent SDS with pH of 8.3. Importantly, the gel operating pH for Tris-Glycine system is 9.5. So when your protein is migrating through a Tris-Glycine gel, it is experiencing this 9.5 pH throughout the gel run. Therefore, this is a very alkaline gel operating environment for your protein. The common ion in the system is Tris, which is present in the gel as well as the running buffers. Now, the Bis-Tris system includes Bis-Tris and chloride ions in the gel buffer, which has a pH of 6.4. The running buffer ions are Tris, MES, or MOPS, and ionic detergent SDS with pH of 7.3. So importantly, the gel operating pH for Bis-Tris system is 7. So this is a neutral pH environment, and when your proteins run through a Bis-Tris gel, they are preserved better because they are not exposed to alkaline or acidic pH environments while electrophoresis. So the neutral gel operating environment helps in getting better resolution as well as preserving the sample integrity. The Tris-Acetate system includes Tris and acetate ions in the gel buffer, which has a pH of 7.0. The running buffer ions are Tris, Tricine, and ionic detergent SDS with pH of 8.3. The gel operating pH is 8.1, which is slightly alkaline, but not as much compared to the Tris-Glycine system. In the next few slides, I'll discuss the different gel chemistries of precast gels and their advantages. As we have discussed already, the Bis-Tris gel chemistry has a neutral pH operating system, which plays a vital role in minimizing protein modification and protein degradation, thereby preserving sample integrity. Since Bis-Tris gel chemistry is comparatively less reactive with acrylamide, the Bis-Tris gels have a shelf life of up to 16 months at room temperature. A Tris-Glycine gel can't be stored at room temperature because of its higher reactivity and a comparatively shorter shelf life. It is also known for quite some time now that a neutral pH environment during electrophoresis improves protein stability, which enables sharper band resolution and accurate results. This slide shows comparison of the same samples, which are purified protein kinases implicated in cancer, loaded on Invitrogen Bold Bis-Tris Wedgewell gel and Biorad Tris-Glycine TGX gel. Since Bold Bis-Tris gels have a neutral gel running pH environment, the sample integrity is preserved and protein degradation, if any, is very minimal. However, with Biorad TGX gel, multiple degradation bands are detected along with the full-length purified proteins. This demonstrates that the Bis-Tris chemistry with neutral pH minimizes degradation during gel runs. The Tris-Acetate chemistry is specialized for optimal protein separation and transfer of high molecular weight proteins up to 500 kilodalton in size. If you are working with high molecular weight proteins like EGFR, BRCA, or some membrane protein, this is an ideal gel chemistry for such applications. Migration charts are provided by all precast gel chemistry vendors, and they are a great resource in deciding which particular gel chemistry or gel percentage within a chemistry will be optimal for your protein of interest. Since a Tris-Acetate gel can separate the high molecular weight proteins better, it is able to move these bigger proteins more towards the center of the gel, and they are not all stacked at the top near the stacking gel, which significantly improves the transfer efficiency. The image on the right shows detection of EGFR receptor at reducing lysate concentrations from left to right. The new page Tris-Acetate gel on the top shows significant better transfer efficiency and sensitivity of detection compared to Tris-Glycine gel at the bottom, which is not an optimal chemistry to detect high molecular weight proteins. On the other hand, if your interest is in detecting low molecular weight proteins and peptides, you should use the Tricine chemistry that is optimized for separation and transfer of low molecular weight proteins. As demonstrated in the migration chart, the Tricine gel enables the smaller molecular weight proteins like caspase subunits to be optimally resolved closer to the middle of the gel compared to the Tris-Glycine chemistry. As seen in the image on the right, the Novex Tricine gel on the top provides a very distinctive separation of the cleaved caspase 3 subunit compared to the Biorad TGX gel at the bottom. Additionally, smaller molecular weight proteins experience very high blow-through effect during transfer process, which means that the smaller molecular weight proteins are blasted past the PVDF or nitrocellulose transfer membrane and are lost, resulting in non-optimal transfer condition. The ion concentration is significantly higher at the bottom of the gel near the diaphragm, and therefore this area experiences disproportionately high current during transfer, which causes this blow-through effect. The Tricine gel provides separation of this smaller molecular weight protein closer to the middle of the gel, thereby preventing the blow-through effect to a much greater extent, and which results in improved transfer efficiency as seen on the Tricine gel over here. The Tris-Glycine chemistry is the most widely used gel chemistry and is used for a broad range of protein separation. It does have high alkaline operating pH. Since Tris-Glycine is a very reactive gel chemistry that results in protein modification due to chemical alterations and reoxidation of amino acids, as discussed previously, it can cause band distortion, loss of resolution, or increased background during Western detection because of protein degradation bands that are recognized by specific antibody against the target protein. We understand that Tris-Glycine chemistry has its limitations, and we strongly recommend that it should not be used for every application in your lab. The one-size-fits-all approach is really not optimal or efficient when it comes to selecting a precast gel for your research application. Having said that, we have worked hard to overcome some of the limitations of Tris-Glycine chemistry while utilizing its inherent advantages. The Novex Wedgewell Tris-Glycine gel is designed to provide excellent band resolution, sharpness, and staining efficiency compared to Biorad TGX gel. You can see better separation of multiple protein bands in lysate, and the Novex TG gel also shows relatively less protein degradation of purified protein. The protein bands are sharp and straight and does not show the smiling of bands, particularly in the first and the last lane, which is very visible in Biorad TGX gels. So with all this background information on gel chemistry and sample preparation, we can answer this question of which gel is right for your specific research application. This table provides a quick overview. If you're working on applications like post-translation modification, which includes tyrosine phosphorylation, or serine phosphorylation, or threonine phosphorylation, or you're working with downstream mass spec applications for your target protein, or your target protein is in low abundance, which requires maximal sample integrity, then you should choose the Bis-Tris gel. Bis-Tris gel will not only help with preserving sample integrity and reducing degradation, but it also minimizes protein modification during electrophoresis that can have significant impact on downstream mass spec applications. If you're working with high molecular weight proteins, anything 200 kilodalton and higher, the recommended gel is the new PAGE Tris-Acetate gel that will provide optimal separation and transfer of high molecular weight proteins, thereby increasing sensitivity of detection during Western blotting. If you're working with low molecular weight proteins, anything 30 kilodalton and under, the recommended gel is Tricine. The Tricine gel that is designed for optimal separation and transfer of smaller molecular weight proteins and resolve these proteins towards the middle of the gel, thereby preventing or minimizing the blow-through effect during transfer that impacts the small molecular weight proteins. So if your application requires the use of non-denaturing condition or native separation, we offer native gel options in Bis-Tris, Tris-Glycine, and Tris-Acetate chemistry. We also offer options in Bis-Tris and Tris-Glycine chemistry for isoelectric focusing and two-dimension gel electrophoresis experiments. For protease activity application, we also offer the zymogram gels. Our website has some very useful resources that you can download, including technical handbooks for electrophoresis, transfer, and detection. There is also an interactive gel selection tool that will help in easily deciding the right gel chemistry for your research application. Thank you for attending this webinar today and providing me the opportunity to highlight some of our key product advantages. We do have time for a couple of questions and we'll be happy to answer them now. I will respond to all the questions we have received today via email in interest of time if I can't get to all of them today. Thank you.
Thank you, Dr. Tomar, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click on the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, can customers use their electrophoresis buffer with Tris-Glycine precast gel from Thermo Fisher Scientific? Thank you.
Yes, so the answer to that, if I understand this correctly, the question is, with precast gels, can you use home-brewed gel running buffer? So the answer to that is yes. Obviously, you have to be mindful that you are using the right buffer for the right chemistry. So for example, Tris-Glycine gels, there is Tris-Glycine running buffer. If you home brew that buffer, that's absolutely fine. But if it is a Bis-Tris gel, you have to make sure that you are using the Bis-Tris running buffer in that case. So all of our precast gels, we have ready to use running buffers, but at the same time, they are compatible with the home brewed running buffer as well, as long as the right recipe is used.
Great, thank you. The next one is, sometimes the protein bands in some of my gels are not straight and well-defined, but are irregular or wavy. What can cause this? So yeah, that's a good question. As
as we discussed today, right, the sample prep, again, is an important consideration for any successful gel electrophoresis or an ideal gel electrophoresis run. So having your bands wavy can depend on multiple contamination that can be introduced during sample preparation. DNA is known to cause bands to be in a V shape, or salt concentration would cause a band to appear a little bit squiggly. It's like we discussed today, it is important to make sure that such contamination are either removed or kept at a minimal to avoid such artifacts during gel electrophoresis.
Thank you. Next one is, can I run reduced and non-reduced protein samples on the same gel?
Typically we do not recommend doing that. For some reason, if you have to run reduced and non-reduced sample on the same gel, it's important that you don't run them side by side. The reason being that there can always be a little bit of spillover, and then the reduced sample can compromise the integrity of the non-reduced sample. So if possible, prevent running them on the same gel, but if you have to, then definitely don't run them in side by side in next lanes to each other.
Thank you. Next one is, I sometimes get V-shaped protein bands while running my gels. What might be the reason behind that?
This is, again, a question similar to the one that I just answered about not getting bands that are sharp, straight, and flat. A V-shaped band in particular is caused more by DNA contamination as opposed to salt or other contaminants. So if they are, typically very V-shaped, then it has to be because of a DNA contaminant, most likely. So, you should either sonicate your sample to shear the DNA that might be present, or you can do an ultracentrifugation step to then, eliminate the DNA from your solubilized proteins, and that should help improve your band quality.
Thank you. Next one is, sometimes I get a blue wave with my gel loading dye near the bottom of the gel. What causes that? It happens especially with precast gels. It does not happen that frequently, but it does happen occasionally. And the prominent reason for that is your sample buffer can get diluted for multiple reasons. One of the reasons can be while preparing fresh 1x sample buffer, if for some reason it's less than 1x, right? So for example, if you add a little less buffer so that it's really not 1x, so that can result into a dilute sample buffer during electrophoresis, which can cause the wavy different. Or in some instances, customers do reuse the gel running buffer. The problem with reusing the gel running buffer is when you have fresh running buffer, your ion concentration is 1x, and when you run, everything is in good proportion. Now, when you are reusing it, the buffer ions are depleted. So you don't know to what extent they are depleted though, right? And it can vary between every run. Now, with a depleted sample buffer, your electrophoresis conditions are not optimal, and towards the end of the run, that can cause some of the lanes to run faster compared to others, which results in this wave, which can be seen through a diaphragm. So I think to avoid that, always use fresh running buffer, and while you are diluting your running buffer, pay close attention that it is truly 1x and it's not diluted in making your buffer process. Very good, thank you. I would like to once again thank Dr. Tomar for his presentation. I would also like to thank Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through September of 2019. Please share with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.